Hi, welcome back to the Sound of Science. Today we're going to be talking about cells, specifically cell theory, how we get our knowledge about cells, where it comes from, and the individuals involved. So here is your heads up. You need to make sure that you know the individuals and their contributions and roughly about the time that they were doing their work. You also need to make sure that you know all parts of the cell theory. Those are the things that you're going to need to memorize from this. So hopefully that will help you as we move forward. Okay. So to start, we're going to start talking about this guy, Robert Hooke, who sounds like he should be a pirate, but he's not. Robert Hooke starts out in 1665 and he is working in England. The picture there that you see is what we think he looked like. His only portrait was lost, which I'll explain in just a minute. But Robert Hooke is working at the Royal Academy of Sciences in London, and this is very prestigious, sciencey place to be working. He is using an early version of the compound microscope. Compound microscopes have two lenses, so they have reasonable magnification and reasonable resolution. Um, he essentially looks at a piece of cork at one point, and the cork looks like it has these little tiny rooms in it. Well, what he's seeing is part of a plant, and he's actually seeing empty cell walls because at that point the plant had already died. This is part of the bark found on the tree that produces cork. So he is, and yes, that cork, the cork that you use on like cork boards. So he is looking at this thin slice of cork. He sees these little, looks like little rooms, and he decides to name them after the cells that monks live in. So monks are kind of like priests, but they don't preach as much. They're more of living together in sort of a monastery commune type situation. Oftentimes they take a vow of poverty. And so monks are not known to have very lavish places to live. Think more of like a college dorm room without anything. Sometimes there's a bed. That's about it. That's about all they have. And so these little squares reminded him of these empty rooms where monks lived. Well, those rooms are called cells. And so he named cells after those rooms, after the rooms in these monasteries. Later, the prison system would also use that same term. And again, it was for the same kind of reasons. They wanted prisoners to spend time in quiet contemplation, and monks spent their time in quiet contemplation. That's why they didn't have a whole lot in their rooms. So Robert Hooke actually is, I don't know if arrogant's the right word, but he's more of a I can do it better than you type of person, more of a one-upmanship kind of mentality. And he actually ends up picking a fight with Sir Isaac Newton, the gravity guy. And so they're both at the Royal Academy of Sciences at the same time. They sort of get into this argument. Robert Hooke thinks that Newton is using theories that he created because Hooke did things other than biology. He worked in physics, he worked in mathematics, and all sorts of subjects. And so Newton, when Hooke died, we won't know for certain that it was Newton that did it, but it's theorized that maybe Newton helped himself to some of Hooke's possessions, including the only portrait we actually have of Robert Hooke. So, what you see today, the paintings that are made, the portraits that are made today, are based on what we think he looked like due to descriptions of him that were left behind. So, Robert Hooke, remembering that he is very much I can do it better, is literally writing the book on microscope. So this brings us to this guy right here, Anton von Leeuwenhoek. In Holland, he is a cloth merchant. Think like Joanne Fabrics. He uses this thing called a jewelry loop or jeweler's loop. Basically, it's a simple microscope. It's like a magnifying glass, and he uses it to see the thread count in the cloth that he's trying to sell. So Leeuwenhoek is not very impressed with the jeweler's loops that are being made. So he decides he's going to start making his own lenses, and he does. And he gets really good at it. He's actually a very skilled lens maker. He ends up creating this elegantly designed, simple microscope. That's what's pictured above. The guy is actually looking through it at something on the specimen stage. That little tiny hole that you see is actually where the lens is. And so Leeuwenhoek is like a kid with a toy. He is looking at everything. He looks at pond water, he looks at teeth scrapings, he looks at blood, he looks at some other bodily fluids, he looks at everything. And it's the pond water that really gets his attention. When he looks at the pond water, he sees these little tiny creatures moving throughout the water, and they're alive. This is the first time we see living cells. Well, Robert Hooke has named cells, and he's literally written the book on microscopes. And so Leeuwenhoek sends him drawings and says, "Look what I see in the pond water here." Robert Hooke, remember, he's a scientist. He might be like, "Oh, yeah, whatever guy," but he's a scientist. And so he goes, he gets some pond water, he looks under his compound microscope, and he sees nothing. So he writes back to Leeuwenhoek and he says, "Thank you for your interest. I appreciate the drawings. However, I don't see them." So Leeuwenhoek sends him more drawings. And Robert Hooke goes out and he gets more pond water and he looks and looks and looks and he sees nothing. So he sends back another letter to Leeuwenhoek and he says, "I really think these animalcules of yours, maybe they just live in Holland. They're not in England." And Leeuwenhoek sends him even more drawings. And at this point, Robert Hooke is like, okay, either this guy is crazy or I am just not doing this well enough. So he gets every candle, oil lantern, everything he can think of, and he puts it in his lab and he puts as much light as he can into the microscope. And suddenly he can see them. He can see these animalcules. What we now know today are bacteria or protist. He sees them. And this is the first time we see a living cell. So Leeuwenhoek is actually the one who gets credited, even though he's not a scientist, for the discovery of living cells. Okay, this brings us to almost a 200-year jump in the future to Theodor Schwann and Matthias Schleiden. Both are German. They're living on opposite ends of Germany. One is a biologist, that's Schwann, and he's actually studying animals. He actually used to go collect samples from butchers. Um, just remember, he's the animal guy. His name is Schwann, which sounds like swan. We also have the Schwann cells, which are nerve cells in the brain that are named after him. He did lots and lots of work on vertebrates, on animals with backbones. Matthias Schleiden, about the same time, is doing the same kind of work on plants. He's a botanist. And ultimately, they end up communicating with each other through mail and determine that animals and plants are made of cells. Well, and at that time, that sort of led to the leap of all living things are made of cells, which is important. That is one of the characteristics of living things, is that it has to have at least one cell. So Schwann and Schleiden are the ones that kind of come up with that part of the theory. And it gets added to the depth of knowledge about cells or about cytology. And then finally, we've got Rudolf Virchow. And Virchow is just a little bit past the other Germans in this group, Schleiden and Schwann. He's in 1855. He is a physician, he's a doctor. And he is really interested in finding a vaccine for tuberculosis. Sadly, Virchow was never able to find a vaccine for tuberculosis. He worked very hard. He creates protocols for the development of many vaccines. He figures out that you don't have tuberculosis just randomly appear, that you have to have a bacteria to cause it, or you have to have something that leads to the illness. And so he creates this whole germ theory protocol, which is really amazing. And we will definitely address that at a later time this year. But for the moment, what you need to know is that Virchow is the one that determines cells have to come from other cells. So, true to the nature of science, all of these scientists added information, and that information gets built into the theory, all of it. You don't just get to say, "Oh, I don't believe this part of the theory." all of it has to be included. And so the cell theory is all living things are made of cells. Cells are the basic unit of structure and function for living things. And new cells are produced from existing cells. In other words, cells have to come from pre existing cells. So all of that is part of the cell theory. But as you look at this and you realize that this is all very important for biology, it's very important for science. In addition, you need to understand too that the story of the cell theory is not just about the nature of science, not just about the study of cells or cytology, but it's also about technology. So we learned more information as our technology improved. Simple microscope, a single lens, compound microscope is developed, multiple lenses that allow us to see it more clearly and higher magnification. And then in the 1950s, we discover that if you take a beam of electrons, the same ones that are zipping around the atom, and you shoot them at the surface of a specimen, they'll bounce off in a pattern that computers can recognize. So we needed not only the ability to control electrons, but we also needed the ability to use a computer. And so these electron microscopes are capable of showing us incredible images. And that's what actually we see here. The purpley type image, that's what you would see in a light microscope. The image of the butterfly with its proboscis just sort of looped around, that's actually a scanning electron microscope. That gives us a vision of the surface. The transmission electron microscope shows us internal features. And what I want you to understand is that the magnification is a huge improvement. If you look at the picture all the way to the right, that's a mitochondria. You can actually see the structures inside the mitochondria, whereas with a light microscope, you really are only seeing the nucleus, and that's about it. So something to keep in mind as you think about cell theory, this demonstrates the nature of science. All the scientists were working, all of them are working on their own thing. They're sharing that information with each other. And this gives them the ability to build a cohesive theory. That's it for cell theory. I look forward to seeing you next time.